Hello, everybody. My name is Mario Diaz. I'm from Universidad de La Laguna, the Canary Islands in Tenerife, in Spain. The presentation I am going to give today is integrated in a more ambitious and multidisciplinary project aimed at the study of human breast cancer and metastasis. In particular, I will focus on the development of multifunctional tamoxifen derivatives, which behave as antiestrogen, fluorescent molecules, and also as photosensitizers. The importance of tamoxifen is that it is nowadays the most widely used compound for the treatment of breast cancers, in particular in the estrogen receptor positive type of breast cancers. The incidence and mortality rates for breast cancers are the highest amongst all different types of cancers in both sexes at all ages. The fact that mortality is so slow comparatively with the incidence lies in the fact that most of these type of cancers can be treated by compounds named anti-estrogens. One of the most and best representative is tamoxifen. The molecular subtyping of these metastatic breast cancers indicate that most of these types of cancers, between 60 and 80%, are positive for estrogen receptors. Estrogen receptors are a group of cytoplasmic proteins that belong to the superfamily of nuclear receptor proteins. In the case of estrogen receptors, they are activated by binding to the natural hormone 17-beta estradiol, which crosses the plasma membrane of breast cells and binds the estrogen receptors in the cytoplasm to form the hormone receptor complex, which upon activation translocates into the nucleus and binds to specific sites on chromatin that contains estrogen receptor element sequences, which finally will control a number of processes, including proliferation and differentiation of cells. The usefulness of tamoxifen in the treatment of metastatic breast cancers lies in the fact that tamoxifen can associate and penetrate into the ligand binding domain of the estrogen receptor so that estradiol cannot activate and form the hormone receptor complex. By doing so, it prevents the activation of sites in the chromatin containing the ERE sequences. The proven efficiency of tamoxifen as antiestrogen is reflected in the fact that it is the most common strategy used for the prevention of recurrence of breast cancer in women after having been submitted to surgery. However, despite the successful usage of tamoxifen as coadjuvant therapy in the treatment of breast cancer, evidence accumulated over the last decades have revealed that tamoxifen is not devoid of side effects, ranging from alterations in the biology of the endometrial and womb cancer to lymphedema. The most frequently reported side effect was hot flashes, and the most worrisome gynecological side effect was a 2 to 3 fold increase risk of endometrial cancer in postmenopausal women. Anyway, nowadays it is considered that despite its side effects, the benefits of tamoxifen in controlling breast cancer or prevention of its relapse are still without debate. At least in part, some side effects are due to the ability of tamoxifen to interact with molecular structures other than estrogen receptors themselves. In fact, some of these effects are mediated by actions that are independent on transcriptional activation or modification of the proliferation programs of breast cells. In an attempt to get a deeper insight into the molecular targets mediating these genomically and non-genomically mediated effects of tamoxifen, we developed in 2013 the first fluorescent tamoxifen derivative, which we call FLTX1, and was formed by the covalent binding of tamoxifen with a low molecular weight fluorescent dye called MVD. The molecular and spectroscopical properties of this structure reveal that it could be excited at the expected molecular excitation of MVD around 460 nanometers with a high quantum yield emitting light at around 530 nanometers. This allowed us to explore by means of confocal microscopy the presence and the colocalization of FLTX1 with different targets within the cell. We see here that we have a robust binding of FLTX1 within the MCF7 cells, which are human breast cancer derived cells with estrogen receptor alpha. The quantification of colocalization demonstrated that between 40 and 70% of the FLTX1 targets were labeled by the two compounds, meaning that between 30 and 60% were non-ER mediated targets. We further explore the potential labeling by FLTX1 of other intracellular targets. The first of it was the potential to colocalize with estrogen receptor beta in MCF7 cells. We observe a consistent colocalization of FLTX1 and estrogen receptor beta in the perinuclear compartment of these cells. Interestingly, FLTX1 was also capable of labeling mitochondria. And this was interesting because of the effects of estradiol on mitochondrial dynamic has been reported several times in the literature, although it has never been visualized. Something that was very interesting on the properties of FLTX1 was that it was also capable to label targets at the plasma membrane. Some of these targets are supposedly responsible to many of the non-genomically mediated effects of tamoxifen. We next explore the pharmacological properties of tamoxifen by using pure preparations of estrogen receptor alpha. We found that FLTX1 was slightly more potent than tamoxifen in binding estrogen receptor alpha. And this is important because not only the modification of the molecular structure of tamoxifen did not affect its capability to bind estrogen receptor alpha, but also that the complex exhibited a higher affinity for the complex. And this was demonstrated and explained by the fact that using bioinformatical tools, we were able to demonstrate the presence of an additional binding within the ligand binding pocket. As mentioned before, some of the side effects caused by tamoxifens are genomically mediated, and this occurs through activation of estrogen receptors. In this part of the slide, we can see how tamoxifen in a range of concentrations can induce a significant activation of transcriptional activity, a finding which is not observed for FLTX1. And this is very important because it is expected for a proper selective estrogen receptor modulator not to have any estrogenic response, especially in target tissues prone to develop estrogenic effects. On the other hand, when we explore the anti-estrogenic effect mediated by blocking transcriptional activation, we see that both tamoxifen and FLTX1 are potent inhibitors of the response to estradiol. It means that the growth of cells, like in the case of mammary cells, is dependent on the presence of estradiol. The presence of any of these antiestrogens are sufficiently potent as to block the proliferation of metastatic cells. This is something that was somehow predicted by this pharmacological study of both antiestrogens. But perhaps the most striking and unexpected effect of FLTX1 was the fact that it was lacking the trophic effects of tamoxifen in uterine tissues. As shown here in the mice model of uterotrophic assays, where estradiol and tamoxifen provokes a considerable increase in the weight and size of the uterus, FLTX1 did not show any of these responses. This was paralleled by the fact that FLTX1 was unable to stimulate the expression of the PCNA, which is a marker of the proliferative activity within the uterus. So FLTX1 was totally devoid of the uterotrophic effects of tamoxifen. This pushes FLTX1 as a potential heir of tamoxifen, since it is capable of binding estrogen receptor alpha with a high affinity. It is also capable to displace estrogen receptors and act as a anti-estrogen in breast cancer cells, but lacks the effects in the uterus. One of the most striking properties of these novel fluorescent derivatives is the appearance of emerging properties that were not present in the original compounds. This is the case of FLTX1, which, above a certain level of excitation energy, develops amplified spontaneous emission. This consists of a reduction in the amplitude of the emission spectra, which becomes narrower and centered around approximately 550 nanometers, which is in the green emission band. The important is that this is a very high level of emitted energy and with an efficiency of about 29%, which is the highest value of efficiency reported for any laser dye reported up to now. This is a proof and demonstration of how this amplified spontaneous emission occurs. FLTX1 compound is here inside the cuvette, and you will see that when the the pump power is increased above a certain threshold, a green laser emission occurs with very high energy. This laser emission still occurs in the absence of mirrors and is only due to the reflections inside the cuvette walls. This amplified emission is observed not only in vitro in a cuvette, which is irradiated, but also is observed when applied to tissues, like in this case in the uterine tissues, where this emission behaves more or less the same way, but with the difference that these reflections occur in a random way in a different configuration. We thought it would be a good idea to incorporate a new functionality to the fluorescence derivatives. The idea was to couple the laser emission of FLTX1 to the generation of reactive oxygen species in order to have a potential molecule for photodynamic therapy. We found amongst the different potential photosensitizers that rose bengal could be a good alternative for this purpose because the absorption spectra of this compound was overlapped to the emission of FLTX1. So we could have rose bengal absorbing in the more or less the same band that FLTX1 is laser emitting. So therefore, we proceeded to the synthesis of a new compound that we named FLTX2, which is based on FLTX1, but incorporating this new functionality as at the end of the molecule as a moiety bound covalently to the MBD. We choose a proper linker because of the efficiency of the energy transference from the fluorophore to the rose bengal is critically dependent on the size. The optical properties of the novel compound were quite approachable to the expected transference of energy through a Foster resonance electronic energy transfer with a very high efficiency of about 95% in the experimental measurements. The fluorescent behavior of the FLTX2 revealed that the emission at about 600 nanometers, which is the wavelength of emission from rose bengal in cells labeled with FLTX2. And we also detected that the emission at the expected wavelength of FLTX1 was negligible compared to the emission of the rose bengal. This indicates that most of the energy absorbed by the FLTX1 is transferred in the FLTX2 to rose bengal and is emitted accordingly. We next explore the pharmacological properties of FLTX2, and we realize that the incorporation of a bulky moiety to FLTX1 did not affect the binding affinity for the estrogen receptor. On the contrary, it seemed that FLTX2 was slightly more potent than tamoxifen and equipotent to FLTX1. The similar affinity for the estrogen receptor of FLTX1 and FLTX2 may be explained from the results of molecular docking studies, which reveal that both fluorescent compounds can bind the same residue inside the ligand binding domain. Although in the case of FLTX2, a new interaction appears between the rose bengal moiety and an aspartic amino acid situated outside the ligand binding domain. At this point, we wanted to check whether the newly developed FLTX2 molecule was capable of generating reactive oxygen species in response to laser stimulation. For this purpose, we use a method based on the absorption of superoxide anions by a compound named nitroglutathione, whose absorption spectra changes according to the reaction with these reactive species. We could observe a irradiation-dependent increase in the absorption in the wavelength of about 600 nanometers that corresponds to the maximal absorption of the oxidized NBT. You should recall that this same wavelength was the emission observed for FLTX2 in cell cultures shown previously. So we next tested for the ability of irradiation to cause any change in the viability assays of cell cultures in the presence of the different fluorescent derivatives. Here we can observe, according to the method by trypan blue exclusion, that irradiation of FLTX2 causes a dramatic increase in the number of cells that show this blue color indicating cell death. We observe that non-irradiated cells behave more or less the same with similar values of viability, while only in the case of irradiated FLTX2 we get a dramatic reduction in cell viability. Also, as a demonstration of this effect, we detected that, as in the case of in vitro experiments, irradiation time was also associated to the viability of cells. So the longer the exposition at the lowest concentration, the larger the number of cell death and reduced viability. In summary, the set of results that we have presented in this study has allowed us to design and to validate a multifunctional derivative of tamoxifen. This derivative behaves as antiestrogen as it is capable of binding estrogen receptors with similar affinities than tamoxifen alone. But the presence of the fluorophore in the molecule allows not only the excitation of the molecule, but also the generation of amplified spontaneous emission in the form of a laser emission, which is transferred through FRET to the rose bengal, which behaves as a photosensitizer. Therefore, the excitation of this molecule allows a massive generation of reactive oxygen species in the same cells where estrogen receptors are bound to the compound and therefore are useful for the treatment of highly expressing metastatic breast cancer cells. Finally, I would like to thank all the members of the different departments that have participated in the project, without whom this multidisciplinary project would not have been so promising. Thank you so much for honoring me with this Best Researcher Award on oncology and cancer research.